All righty, good afternoon, good afternoon. Try to get everybody in. All right, good afternoon. Have your worksheets handy. We got a lot to do today. Try to get this finished and submitted. And then I want you to download that review worksheet. Good afternoon, good afternoon, good afternoon. All righty. Hello, Christopher. Are you new to the class? Are you okay? All righty. Well, welcome. All right, we are in Canvas. Okay, let me come out for your benefit. I'm gonna come out and show you where we are. We are in Canvas, and we're in the unit for DNA mutations. All righty. Okay, so there was a worksheet with this. So we're talking about how to use the coding wheel or the table. Either one. I'm using the table with this activity, and we're talking about mutations. So if you go from there and hit next in Canvas, you'll come to this worksheet. And if you download that, that's what we're working on today. We're actually halfway finished with it. And we are here. All right, so we've been talking. So again, folks, we're trying to get finished with this so that you can upload it. So stay with me, stay with me. Some of you might have gone ahead and answered the questions, which is good because everybody present should, with the exception of Christopher, um, should be able to upload this worksheet today. If your body's in the room, you should be able to upload because you've been following along, doing your work like you're supposed to, and putting in your answers. And if you can't, you should be asking a question as to, I don't understand the answer to this. Yes, Ethan, you can do that. And I'll check that when I take a break. Alrighty, so everybody should be here. And we stopped here talking about okay, let me back up just a little bit. So over here we were talking about the two types of mutations. I'm going to review hopefully so everybody gets in. All right. So the first one that we talked about was point mutation, and that went along with our little cow and human insulin translation. And so we looked at the fact that insulin or diabetes is the disease caused by a point mutation, meaning that there is a particular set of nucleotides that have been rearranged on the strand, creating a different amino acid. So you can isolate the one amino acid that is causing the difference. That's point. On a silent mutation, if we look at the chart again and notice that there are 3, sometimes 4 ways to get the same amino acid. And if we change it in that way and get the same amino acid, there's no change in the expression of the protein. So therefore, the mutation is silent. Okay? Which is not the case in diabetes. And then we looked at sickle cell, and we looked at, they were asking you if you compared it to point or silent. We decided that it was a point mutation because there was just one difference. If you're looking at these two, the set right here is different. The top is normal, the one on the bottom is different, and it created a different amino acid, which made the protein behave in a different way, one that creates hemoglobin that doesn't carry the sufficient amount of oxygen. And so it doesn't work like it's supposed to. So it's non-functional. Again, if you have to, and these are your worksheets, if you have to highlight, underline, you do that. But it says it is one, where is it now? Okay, here it is. The shape changes it and it becomes less functional or not functional at all. Okay, so that's the case with the hemoglobin. It is less functional. They don't die right away, but it causes the red blood cell not to function the way it's supposed to. All right, so then we were looking at frame shift, and we translated. So again, you have to translate the normal RNA so that you get the RNA codes. And then you had to go look for the amino acid on the chart. And then what we looked at was if this is the DNA, and DNA all of a sudden has an extra A inserted in there. Okay, that means that now we're coding, instead of coding for serine like we were up here, now we're coding for lysine down here. So hopefully you filled all of those in. And then the question was asking, why is this cause more problems than a point mutation? And our answer was what? Somebody tell me what the answer was.
That point changes one, but farm shift changes all of them.
Okay, everything after this change is going to be changed because now that we're reading them in sets of 3, the sets of 3 have changed. So notice up here we had CGA, now we've got TCG because it's shifted over one. And then up here we have GGT, now we've got AGG. So everything after that one insertion is going to change, and you're going to create an entirely different chain of amino acids, therefore a different protein. All right, so that's what you should have written in that box. Everything after the change, after the insertion, and those are nucleotides. Everything after that nucleotide insertion will change how all of the triplet codes after it. All right. So now we're going to look at cystic fibrosis, which is a disease that's caused mucus to build up in the lungs. The underlying cause is a mishap-shapen protein in a cell, which interferes with the transport of chlorine. That's what its job is supposed to do. All right, researchers have identified 2 frame shift mutations associated with this disorder, one associated with the nucleotide insertion, so that kind of looks like the one up here, and another caused by a nucleotide deletion. Okay, so that means that in one case, it looks like the one up top. But in the next case, let's say, for example, instead of that T being there to go with the next triplet, that T disappears altogether. So that nucleotide is not there to help represent the other sequence that should have been there. All right. All right. So why can both deletion and insertion mutations have the same, cause the same disease? Because it interferes, and I'm going to tell you what it is here. So pay attention, write it down. All right. It interferes with this right here. Okay. Chris, just hang in there for a second, and I'll back up in a minute. Okay. Just download the worksheet, and then I'll backtrack because we are. Did you take biology at all? You did, but you weren't on DNA yet. Yeah, let me let these other folks in while you're answering. All right. Folks, y'all late. Y'all late. Okay. All right, we are on the worksheet, trying to get this finished up. And like I said, Chris, I don't expect you to turn this in, um, but everybody else does. But hang in there for a sec. All right. So it affects the protein. It continues to misshape the protein. Yeah, you can say that. That's an interesting way to put it, Ethan. Ethan said, and again, folks, when y'all send stuff in the chat, send it to everybody and not just me. All right. He said they both mess up the chlorine transport. Okay. Both the deletion and the insertion still lead to the protein not being able to a protein that does not allow the transport of chlorine. Okay. All righty. And it's similar to the way we were talking about there's more than one set of amino acids. So I'm sure if we continued the chain of events, it would show that some of the other proteins are going to code for similar amino acids. So it will, aside from the first one being off, it'll still cause the same disease. Okay. All right. So that's cystic fibrosis. You need to remember, keep track. You have a little notepad where you're writing these things down. Number one, you wrote down that diabetes is a point mutation. You're writing down that sickle cell is a point mutation. And now you're writing down that cystic fibrosis is a frame shift mutation. I keep track of all of these. All right, we have another one, muscular dystrophy. All right. All right, so Christopher, I'm going to go back just a little bit. What we have, let me go back to, hold on, I'm going to close this for a second and go back to, and everybody pay attention because remember, you have to do the benchmark when you come back. So this review is good for everybody. Okay, let's start with this one. Okay, so Chris, if you go back to the worksheet that says protein synthesis codon, this is where we started looking at it. So a DNA molecule, so it gives you a synopsis up here at the top. A DNA molecule is a strand of nucleotide bases, and when the RNA comes to read that information, it reads it in sets of 3. And so we call that set of 3 bases a codon. And every codon codes for 1 amino acid. And the amino acids are linked together like pearls on a strand of like a necklace, pearls on a necklace. And so they make the protein. So think of the necklace as a single protein, and each bead on the necklace as an amino acid. All right, so then protein synthesis, making protein is called translation. So we got the information from the DNA, we transcribe the information by creating these triplet codes, and then we translated it by getting the amino acids and making the protein. So in order to do that, to read the triplet codes, we've got to use this little triplet code reading chart. So nobody expects you to memorize it. Anytime you see this on a test, they will always give you the chart, okay? But they do expect you to know how to use the chart. So they're in a triplet code. You have like these down here to the bottom. There's 3 bases in a row. And so you go first, like for this one, it's a C. So the first base on the side here is C. Second base is also C. So that brings you second row, second column. So that means our answer is going to be in this box. And the last letter is also a C. So you're going to find C, C, C in this box. In this case, all 4 of these code for the same amino acid of proline. Now, the amino acids have also been abbreviated with 3 letters just to be short. But in the full name of the amino acids is on this chart. There they go. All right, so the full names of the amino acids are written out on these charts. But most of the time, they abbreviate them with a 3-letter code. Not to be, and folks, don't confuse the 3-letter abbreviation of the amino acid with the 3-letter base code, okay? So the amino acid is abbreviation for a whole name. The base code is a sequence of 3 nucleotides that we're reading and transcribing to get this amino acid. All right. Okay. So let's get back to our, and like I said, Chris, we're gonna have to catch you up. I have a tutorial time Friday at 1:30. If you can pop in to the Zoom, and I can kind of spend more time to explaining this. 1:30 Friday. All right, back to this. All right, so we're on muscular dystrophy, and then a lot. This has been kind of popular. A lot of the TV stars seem to be having muscular dystrophy, and it causes a weakness in the muscles, so they either can't walk, depending on how severe it is. Some are walking with canes, some are in wheelchairs, some can't walk at all. But it's a mutation in the protein, the dystrophin protein. All right, the transcribed gene sequence shown below illustrates a nonsense mutation, similar to what occurs in people with muscular dystrophy. All right, so hopefully you filled that in. So I'll give you a minute in case you haven't. All right, and actually just for the sake of our newcomer, we'll do it together. So we're going to take the GTG, and the RNA strand is going to be CAC. And the TTA. Okay, let me back up, Christopher. You have to remember, and you make a note to yourself on the side, that in complementary bases, G always matches C, A always matches T, except on RNA. So The T will match an A. So there are A's in an RNA strand, but there are no T's in an RNA strand. So like in the next set, when we have a T, we can say A, but then we have A's and we have to say U because U is the substitute on an RNA strand for thymine. There is no thymine in an RNA strand. So we have CAC, now we have AUU, and then we're going to have CCA, and then UAA, and GCC. So everybody should have written that down. I'll say it again. We've just translated the DNA into RNA. So we should have CAC, AUC, CCA, UAA, and GCC. Okay. All right. Thank you, Ethan. Ethan's been transcribing for us in the chat. All right, so our next step now is to go find the amino acids that go with this. So we look on the side, we have C on the left, we go up to the top, that's A, that's third column. So our answer is going to be in that second row, third column. We're looking for, so that's going to be histine. All right, A is the third row, U is the first column, and U is going to be the one at the top. So that's going to be ILE, which is isoleucine. Thank you, Ethan. Ethan's got all the answers in the chat. Good, good. We're missing one. All right, you got this. Okay. CCA is proline. Good, good. And now our UAA. And notice that says stop. And then our GCC. Okay, it's alanine. All right, now it's nonsense because you can't have a stop amino acid in the middle of the chain. So it's still coding, but if it tells you to stop in the middle of it, then it's not going to really make the rest of them. The chain is going to break there and it'll start a new chain. So that'll create a short protein, which is probably going to be non-functional. Okay. All right. So it says propose a definition for nonsense mutation. And then you actually have a definition in your vocabulary. Nonsense mutation. What would you say is a nonsense mutation? I'm gonna close this for a minute and go back over here. You don't have to make one up. Okay, that's a good way to put it. It won't work if you stop. Okay. Okay. All right, there we go. All right, a base pair that results in a premature stops and the protein is non-functional. So you can write that in that space.
What do we write in the space of the stop?
You're going to put stop in the box. Where you're putting this answer, the question asks you, propose a definition for nonsense mutation. So I'm not asking you to propose one. I'm just going to ask you to come back here and copy the definition. You don't have to make, although Ethan nicely put it that it won't work if there's a stop in the middle. And that's a good answer. But it creates a premature stop. It's not supposed to stop there. It's supposed to keep making the protein, but all of a sudden you're stopping. So it's a premature stop. Thus, it's going to create a short protein that has no function at all. Okay? And I'm showing you where I got it from so that when I move on, you can go back and get it for yourself. All right, the second question says, why would a nonsense mutation be less damaging if it occurred at the end of the gene rather than at the beginning? All right, why do you think?
Because if it's at the end, then it doesn't really have any effect because it was going to stop anyways. But if it's at the beginning, it's just cutting the thing short, and it's just not going to work.
Excellent answer. Excellent answer. Because at the end, it won't disrupt. That's an excellent answer too, Ethan. It won't disrupt any other amino acids. It was supposed to stop anyway. Okay. All right. So I'll let you take a minute to fill in those two answers if you have not already done so. And we're almost done with this worksheet. Okay. Let me get back to the worksheet. All right. Let me see if somebody can answer this. It says in one sentence, how would you express the relationship between a DNA? What's a good sentence? And again, if you answer in the chat, make sure you send it to everybody. What's a good sentence that explains the relationship? And this really is a summary. All of these questions are summaries.
They give us unique features.
All right, you have to use all the words. So your sentence has to use all. So you have to say DNA does this, and how is it
Right.
related to
So
the codon, and how does that relate to a protein?
DNA gives us unique features, proteins.
No, don't say features. It gives us a what? A set of.
Set
Gotta
of
use. Gotta
unique.
use
No,
a vocabulary. You gotta
set of
use these three words.
codeines.
Yes. DNA gives us codons, which tell us to do, how to do what?
How to make protein.
There you go. And that's as simple as it is. DNA creates a message in the form of codons, which translate to making proteins. That's as short as you can get that. Okay. Remember, the DNA is the mastermind. It has all the information in the nucleus. So the DNA has the information for the codons, which translate to getting the amino acids which make our proteins. That's one sentence. All right, they told you this in the second question in one of the videos. If you go back, proteins have different shapes, and the shape of the protein defines its function. So if it is misshapen, then it does not function properly. Okay, then that's the simplest way you can say that in one sentence. If it is misshapen, if the protein is misshapen, it does not function properly. Simple as that. All right, this one I'm going to let you fill in. I told you to just keep a track of all of these as we went down. So just pop back up to the worksheet, record the symptoms, and write down what type. Is it point? Is it I just went blank. Oh my goodness, point, silent, frame shift, or yeah, those are your choices. Point, silent, frame shift, or nonsense. Point, silent, frame shift, or nonsense. Put those there. And I'm gonna give you a few, I'm gonna stop sharing for a minute and give you a chance to fill in those blanks while I do attendance and check something. What was I supposed to check? Ethan, I was got to check your worksheet. All right, you all work on that and I'll be right back. Also, if you finish working on that, download that review guide. We're going to do that next. Because remember, when you come back from break, you have to do the benchmark. So we're going to, and this will help. So all of this will be on the benchmark, not all, it's not the whole test. Remember, benchmark covers everything that the course was ever supposed to be about. All right, let me do attendance, stop talking, let you work. All right, and you should be able to turn this worksheet in as soon as you've finished with it. So if your body is present, you need to turn it in except Christopher. Everybody else is expected to turn in a worksheet.
What'd you say?
No, you don't have to turn it in. Everybody else does. Because they've been here. They have no excuse.
So we finished the
Okay.
worksheet on our
But
own
you
and then
hang on.
turn
I'm
it
gonna,
in.
like I said, yes, please. Yes, please. Because everything else on that page is a review of everything you did at the top. So you should be able to go to the top and put the answers on the bottom. And Chris, like I said, if you can you meet me on 1:30 on Friday in the Zoom? Because then I can go through some things with you. Okay. All right, Brandon, I hope your worksheet is turned in. All right, so again, if your body's in the room, you're turning in a worksheet. Okay, gotcha. And I'm going to talk with Miss Martin about how your grade is being transferred. So um, there will be a point at which you will just start with where we are. Okie dokie. Attendance is done. Okay, so this last question, so you should be able to fill in boxes with 12 and 13. So for 14, it says, based on the evidence for how the structure of DNA is determined, determines the structure of proteins, which carry out essential functions for life through systems of specialized cells. Okay, so we're looking at the picture. The first box is DNA, and it shows that DNA is double-stranded. Um, then we have uh, RNA, which is single-stranded. So the DNA transcribes, it tells you on the side there, its information to a single-stranded RNA. And then the RNA translates that information to make a set of proteins, each of the amino acids, and then when it's finished making that chain of amino acids, it folds into the protein that it's supposed to be. So that's what that picture is showing you. Okay. All right. So DNA is composed of sugar, phosphate, and what do you call these four things? Somebody tell me.
Amino acids.
No, these four things, A, T, G, and C, are not amino acids. What are
Nucleotide.
nucleotides? Thank you, thank you, thank you. Who said that? Your mysterious voice. You said it so fast, I wasn't looking at who was talking. But nucleotides, these are nucleotides. Okay. Adenine, thymine, guanine, and cytosine are the nucleotides. Remember, our nucleotides get joined together to form, and we're going to go over that on the worksheet in a second. All right, during transcription, so in order to get from the DNA to the RNA, DNA is made into a molecule of what? What's this second little red line called?
RNA.
RNA, which will travel to the what? Where's it going? What organelle is it going to?
The no.
Starts with an R.
The endo. No.
Starts with an R.
Ribosome.
Ribosome. There you go. Isaiah, is that you? All right, so the RNA, so the DNA is going to make a strand of RNA, and the RNA is going to leave the nucleus, and it's going to go to the ribosome, and ribosomes are in the cytoplasm. Okay, they can be floating around. Some of them are free-floating ribosomes. Some of them are on the endoplasmic reticulum. Those are the little dots on the endoplasmic reticulum. All right. Here are three bases called a what? Starts with a C. You put these three bases, so like for example, our GGC, what do you call that?
Chain.
What are these things called up here? This GTG with those, a codon. Thank you, Ethan. It's a codon. All right, 3 bases are called a codon, and they will determine what? What acid? Starts with an
Amino
A.
acid.
Amino acid is added to the chain, and the chain will fold into a, what's that squiggly thing up there in the picture? So our long
Protein.
protein, there you go. All right, changes in the DNA code are called what?
Transcriptions.
No. If it's not making the protein that it's supposed to, then it's called a what? In general, generally speaking, if it's not the protein, it's a mutation. And which mutation is a problem with the hemoglobin? Will cause what kind of anemia is caused by a mutation in the hemoglobin? Not diabetes. It's blank cell anemia,
Sickle cell.
sickle cell. All right, we have successfully answered all questions on this worksheet. Submit, folks, submit. Finish it up, put all the answers in and submit. All right. Like I said, I'm going to jump ahead a little bit. When we come back, after we do the benchmark, we're going to come back and talk about DNA technology and engineering and cloning and some more stuff about DNA. But what I want to do, because you have to do the benchmark test, I want you to skip to the review page. And let's look at that. Now I'm going to go through as many of these answers as possible, but you're going to have to go through and do the rest. So again, in order to prepare yourself for the benchmark. Now again, the benchmark is not a grade that's going in the grade book, but the school does use it to do statistics about how well you're doing, where you're placed, what classes you can be in. And when it comes down to in March and April, they start setting up tutorial classes to prepare you for the benchmark, not the benchmark, the milestone that everybody has to take in April. So they determine where your strengths and weaknesses are from the benchmark. You're going to take another benchmark in March to see how much you've progressed from here to there. So they're going to compare the two scores and then put you in, set up progress learning activities for you to do to help strengthen the areas that you're weak in, so you have a better chance of passing the milestone. All right. All right. So everybody's hopefully has by now downloaded this worksheet. Go back and look at the vocabulary. So step 1, we're practicing. Step 2, go back and look at the vocabulary list that's at the beginning of the module. And that takes us to question 3. So it says draw a model, and you don't have to, and again, I'm not collecting these, so I don't expect you to do this, but I will take you, I'll close this for 1 second. Oh, okay, was that showing? Change to my screen. All right. Let me see something for a sec. Okay, you were looking at it. All right, so I'm gonna close that for a second. So question number 1 wants you to, and you don't have to draw it because you have one already. You have a worksheet that you already did. And it's easier if I do this than try to click back. So you just need to pull up. Why did it jump? pull up your worksheet. So you want this worksheet. Okay, you've already turned this in. So that's what they're looking at. All right, so we use this. It says draw a molecule of DNA and label the following parts. You have already done that on this worksheet. So just go back and look at it. And again, it shows you that it's double-stranded. And notice one side has the circles going down and the other one has these circles going up. So that tells you that they're parallel, but they run in opposite directions. So that's two things that this picture is telling you. It's telling you that all the little circles are our phosphates. All our little houses are our deoxyribose, are our sugars. And then our bases, the things that we've just been talking about. A, and you should have a T on the other side that bonds with A bonds with T or complements T because we call it complementary base pairing. You always have an A with a T for DNA. You always have a G with a C for DNA. And then if this again should have been a T and it should have been an A. Some of you put TT and I accepted that because whoever made this worksheet was supposed to make this a longer box, but it should be a T and an A, and then a G, a C and a G, and then a G and a C again. So the shape gave you a clue as to what it was supposed to be because all the bases are going to have the same shapes. So they fit like puzzle pieces. Okay. So these are the names of our bases. Adenine, so we're so used to using initials. Adenine is A, guanine is G, C is cytosine, and T is thymine. All right, so you don't have to do number 3 because you just need to go back and find this worksheet and you have all the answers for that. All right, comparing DNA and RNA, determine which of the following applies and then write it on the line. So I'm gonna go back to close this and come back to our handout. I really should have 2 pages open. Yeah, let me do that. And then that way I don't have to keep going back and forth. How much time we got? We got plenty of time. All right, give me a second. I'm just trying to 2 pages open so that when I go back and forth, I don't have to make the other one disappear. Okay. All right, so we're on the DNA review page. Okay, so again, and I'm going down the line. DNA is a double helix. Deoxyribose sugar is our little house-looking structure. That's in DNA also. That's what the D in DNA stands for, deoxyribose sugar. Nitrogen base, both. Both have nitrogen bases. Remember, those bases are the A, the G, the C, and the T. Those are nitrogen bases. You have them on both RNA and DNA. The only difference is RNA has uracil instead of thymine, but both. So we have DNA, both. All right, one strand, and is RNA, is single-stranded. We just looked at that on the worksheet that you just turned in. It had the box with the double-stranded DNA, then the red line was a single-stranded RNA. There are three types. That's RNA. There's messenger RNA, ribosomal RNA, and transfer RNA. Write those off to the side. So write RNA on the line, but then off to the side, I want you to write down, and then you abbreviated mRNA, and then there's rRNA, and then these are small letters, the lowercase r, and then a big R, and then tRNA, which is transfer RNA. So write those off to the side. There's three types of RNA. Okay, uracil is only found in RNA. DNA is only found in the nucleus. And both DNA and RNA are called nucleic acids. So that's both. Okay, anybody need me to repeat any of that? All right. Distinguish between pyrimidines and purines. So, see if this is gonna work. You have to I'm trying to figure out where you had that because it's pictures. All right, so so I'll go back and look at these pictures again. So these structures, so again, it takes three pieces to make a nucleotide, the phosphate, the sugar, and the nitrogen base. You put them all three together and they make a nucleotide. Okay, then we have base pairing. Cytosine always pairs with guanine, adenine always pairs with thymine, and they are called complementary base pairs, but that's not what we're looking for. All right, so this is what they actually look like. And somewhere on here, you did have pictures of okay, here you go. Right here. That's what you need. Adenine and guanine are purines, and thymine and cytosine are pyrimidines. So again, you have a purine is always paired with a pyrimidine. Adenine goes with thymine, guanine goes with cytosine. All right, so that's your answer for number 3. And I just answered number 4. Our base pairing rules are adenine pairs with thymine and guanine pairs with cytosine. That's the base pair rule. All right. And if we come back up here. Okay. No, that's not the one.
In 1953, Avery, MacLeod, and Chargaff's rules. Chargaff studied the molecule of DNA in
There we go.
Griffith mice and DNA transformation. Frederick Griffith conducted experiments with two related strains of bacteria. Rough coat bacteria did not cause pneumonia in mice. You can see this in the first column of the diagram. Smooth coat bacteria did cause pneumonia in mice, and the mice died. The reason for this is that the smooth coat actually protects the bacteria from the mouse's immune system. Interestingly, Griffith took this experiment 2 steps further. He then decided that he would try to kill the smooth strain of bacteria first before
Right,
injecting
so
it
write the
into mice.
conclusion down at the
He
bottom
did this in the
as the
third
answer to
column
number 5.
of the picture. The mouse lived. Finally, Griffith decided to mix both the killed smooth strain and the rough strain together and culture them for a few days before injecting them into the mice. When he did this, he found that the mice died. The conclusion that Griffith made was that transformation had occurred. Some unknown chemical in the dead cells was transforming the harmless rough cells into dangerous smooth cells. We now know that this chemical was the DNA from the heat-killed cells that was not destroyed. Bacteria can actually take up the DNA from their environment and use it to make smooth coats, making them more deadly.
Okay, so all you need to write down here is for number 5 is unknown chemical dead cells was transforming the harmless rough cells into harmful smooth ones. That's the answer to your question. All right, hopefully you have your page open. Let me see.
Miescher discovered Avery, Chargaff's rules, Hershey Chase, and blenders. Hershey and Chase experimented to find out if the transforming principle from Griffith's experiments was DNA or if it was protein. Hershey and Chase used a virus called a bacteriophage that infects bacteria. They knew that bacteriophages were made of 2 things. One was DNA, which contained phosphate, and the second was protein, which has sulfur. Which molecule would enter the cell to make more viruses? They labeled the phosphorus and sulfur, each with radioactivity, and observed what happened in 2 experiments. Look at the diagram. Since the protein coat of viruses contained sulfur, the viruses were treated with radioactive sulfur that would bind to the protein but not DNA, and be easily detected with special equipment. This is shown on the left-hand side of the diagram. On the right-hand side, you can see that since DNA has phosphate molecules and not sulfur, the viruses were also treated with radioactive phosphate that would bind to the DNA but not the protein. The treated viruses both infected the cells, and then the cell parts were broken apart in, of all things, a blender. They separated the pieces into groups and determined that the DNA with phosphate was the material that was being injected into cells, and not the sulfur-containing protein. Radioactive sulfur was found on the surface of bacterial cells, and radioactive phosphate was found inside the bacterial cells. The conclusion here was that only the genetic material, DNA, entered the cells and caused transformations.
Okay, so the answer to your question is the infected DNA. Okay. So in other words, what's on the outside coat did not, the phosphorus and the sulfur did not infect the cell. It was the DNA that infected the cells. Later on, we're going to talk about why viruses are not living things. While they have DNA, they have to be in a cell in order to be functional. They can't function by themselves. They depend on living things to read their message. Remember, DNA is a message, so they need a living cell to read their message and create what their message says. But they, by themselves, they don't do anything. All right, so those two. Is the following sentence true or false? Nucleotides can be joined together in a specific order. I don't like the way they asked that question, but it says, oh no, it said must. Okay, must. That's the key. You got to read it properly. Nucleotides must. And that's the key word there. That's wrong. That's going to be false. So we have just done our nucleotides. Some is CCT and some is AGT and some is GAC. Those are random arrangements that create a certain message. So they do not have to be, they don't have to be joined together in a specific order. They are joined in whichever order codes for that message. So it is kind of random. It's specific for a protein, so that's why I said I didn't particularly like this, because it must be in a particular sequence in order to produce the protein that we want. So that is specific. But the general arrangement, they can be a different series. We can have ACC, we can have GGG. They can be joined together in various different ways. But how they are joined together will determine what kind of protein they make. Okay, so I'm not, that question is kind of tricky. Okay. All right, Rosalind Franklin. And that would take us back to this thing here.
In 1953, the structure of the DNA molecule was revealed. James Watson, Francis Crick, and Maurice Wilkins examined an amazing photograph made by Rosalind Franklin, a colleague of Wilkins, using a technique that she developed called X-ray crystallography. Together with this information and the studies of many other scientists, Watson and Crick deduced a model for the structure of DNA. For this, they all received the Nobel Prize. Their model for the DNA molecule consisted of two nucleotide chains that wrap around each other to form a double spiral. The shape was called a double helix.
That's the answer. Double helix. All right, the shape like a double helix. You can also like a spiral staircase. There's a lot of different ways they describe this. A double helix, a spiral helix, but either way, you have to show that it's explain that it's twisted, and it's two strands, and it contains two matching strands. Okay. Okay. What time is it? We have 5 more minutes. All right. From her X-ray, so that kind of answers. They did it from X-rays that Rosalind Franklin created. All right. Is the following sentence true or false? According to the principle of base pairing, hydrogen bonds would form only between adenine and cytosine. That is not true. Hydrogen bonds will form between all bases. Okay, and here again, it says only between. So it's not only between cytosine, and they're not, A goes with T, so that would be wrong in that sense anyway. Okay. There's hydrogen bonds hold those little dotted lines that we had on our diagram, are holding together our A and our T and our G and our C. All right, indicate with a letter the location of the prokaryotic cell. Prokaryotic cells are those that don't have a nucleus. I don't know where this. Oh, okay. It's going to be in the cytoplasm. That's what they want you to. This was a multiple choice question. All right. So they have prokaryotic cells don't have a nucleus, but they do have DNA. So some bacteria, they don't have a nucleus, but they will have genetic material. So it's just floating around in the cytoplasm. All righty. What occurs during the process of replication? The DNA makes a copy of itself in the form of RNA. Actually, no, replication is just it makes a copy of itself. And it's going to, it usually occurs during the beginning of a cell cycle when the cell is about to create a new cell. All right, what does it mean when two strands of DNA are complementary? That means that there is base pairing. They are lined up so that they are the bases are complementary to each other. Is the following sentence true or false? Each DNA molecule resulting from replication has one original strand and one new strand. This is true. All right, what makes DNA replication semi-conservative? Because it only opens a part of it. Okay, it doesn't open the whole thing, so it makes it only replicates certain segments of the DNA at a time. All right, what are the roles? All right, you have these on your worksheet that you did the other day. So let's go back and look at that. DNA structure. Was that in here? No. Oh, that's the one we just looked at. Come on. Okay, you have a worksheet that looks like this. So this is what we call the semi-conservative. Some of the DNA opens up and it makes two new strands of DNA, and then those will break off, and this will close back off. And all of your answers for that are on here. And it's also on the worksheet that you did. So you did a worksheet, which should be on the next page. So if you turned it in, like, okay, here are your answers for those right here. All right, and I'm going to stop right there because we've run out of time. But do go through the rest of it. Like I said, when you come back, we're going to do the benchmark, and it's just where you just have to take the time to study, or just wing it and let them know how much you know from what you don't know. But in any case, you all have a wonderful Thanksgiving break. Be safe, and see you in 2 weeks. And Chris, I'll see you on Friday.